My name is Jeff Wedgwood, and this is the Scientific Evidence for Evolution Class 7. Today is 5/17/2011. Today's talk is about DNA in an advanced form, or at an advanced level. I mentioned to you last time the central dogma of biology. And I also told you that whenever I see the word dogma, it gets my hackles up. Because I'm a skeptic, and I think that we often overstate how much we know. But the idea was that DNA was the storage place where all the information to create a human being was. It's often called a blueprint, and that's completely wrong. A blueprint has something on it which says, like put a wall here, which is the same as, grow a bone here. It's a recipe. You have to follow it step by step. If you look through embryology and watch, you can see the genes being turned on and off in sequence, just like making a recipe. For instance, if you're going to make a good cake, you take the flour and the sugar and the baking powder or whatever, and you mix the dry ingredients first. Then you mix the egg with the milk, and then you add the melted butter to the egg-milk mixture, because if you added the melted butter to the egg, it would cook it, because it's too warm. So it's a recipe. It's the same thing. You have to do things step by step in embryology in order to end up a human. So DNA has a recipe which is extremely complex. That recipe is used to make a copy in the form of RNA, which actually includes messenger RNA, transfer RNA, and ribosomal RNA. So RNA seems to be kind of important. The RNA is used, these three forms of RNA are used to form a protein sequence or a polypeptide. The amino acids are considered monomers. They're single units, and they're connected up to make a protein. And I wanted to just go over that again, if you don't mind. I'd like to. Okay, those are two monomers of amino acids, and the R's stand for the differences between the amino acids that occur in the R's. There may be one with an H, there may be one with the CH, there may be one with sulfur, and so on.
Can you follow such a recipe from the beginning?
Yes, you can. Yeah, you can. Now that we have all of human DNA genome, we can follow it from there to here to here. We also can sequence proteins themselves so that we can see which part of the RNA was used to make the protein. But the point I wanted to get here is, this is where you can have a combination. All you have to do is throw out water, H2O. And what happens when you do that is you end up with these two things bound. Now there is a bond between that and that. And that is just removing that water molecule is what allows the enzymes which read the RNA to make a protein.
Because you double the molecular weight.
Mm-hmm. In this case, if you're doing just a dipeptide, but after you've added 500, 600, 1,000 peptides, Each additional bit is minor.
Are all proteins just one single length?
Some of them have bonds across them. Some of them form the polypeptides, but the polypeptides form polymers. Like hemoglobin has 4 globin molecules on it, but each one is an individual molecule. They're not actually connected to each other. So you've got the 2, but they're connected by hydrogen bonds, which you found in the DNA and RNA. And that really basically was just some kind of a ring structure, which had an OH, and over here some kind of a we talked about that oxygen having some electrons on its outer surface, so to speak, and that means that it's going to attract the hydrogen. And you're going to have, so since this isn't a ring structure, the bonds vary with time, so that you can end up with the opposite, a double bond here and a single bond over there. So that's where the strength of the hydrogen bond comes in between the DNA or RNA nucleotides. Now
Have they ever dug out a methodology that covers more than one evolvement?
Yeah, they have. I showed this last time. This is DNA in mitosis. Whoopsie, hello? Is it back on the screen or not?
Nope.
Let's wait and see. Let's load this up just a tad. Make sure it's in focus. Can everybody see that now? Yep. Good. Need to make it not quite that big. Okay. So as a reminder, mitosis is where a single cell doubles its DNA, stretches out into 2 daughter cells, and ends up with 2 cells that are identical. Notice that during this part, there is no crossover or exchange of genetic material between the different chromosomes. That's very important when you look at meiosis. Meiosis only occurs in the gonads, the testicles, and the ovaries. And if you look here, you start with normal complement of DNA from both the mother and the father. And here you see the crossover. See, there's this one now has a red tip, a blue tip, and a red tip. So when you get to the daughter cells, you don't stop just at 2. You then divide again to make the cells go back down to the normal number of chromosomes, because this is double. So now we have 4 daughter cells. In this case, you can think of them as 4 different eggs in one woman. And depending on which egg she releases at the time that it gets fertilized and becomes a fetus, that fetus will have a possibility of getting different chromosome combinations. And if, for instance, this red represents the father or mother's contribution, which is better than the same thing in that location, which is blue, then this child and this child will have an advantage because they've got that red knob at the top. They will be more likely to reproduce than the others. This is the underlying hypothesis. Anybody have any questions about those two? How they're very different? Mitosis occurs in all cells. Your skin cells are doing it like crazy, trying to keep up with you rubbing off the dead skin layers. But these only occur in the gonads, and that's an important point. I showed you this diagram of the tree of life, so to speak. This one is set up so that it shows where different things happen. Bipedal gait and large brains, 3 bones, middle ear, hands and feet, skulls, body plans, and multicellularity. So this is coming from the past, going to the future, culminating in human beings. Okay?
Completely.
Did you get the sarcasm, the irony in that, or sarcasm? This is so simplified and so directional. It is completely wrong. Or it's not. Some people would say it's not right. Other people would say it's not wrong. It's so off. So, pass this around. This is a picture of a piece of art. Oh, and this was the other tree of life. Vertebra, jaws, digits, amniotic, eggs, hair, placenta, opposable thumbs, going all the way to humans. And I showed you that it's actually a bushy tree, and a whole lot of these things didn't make it. Well, that bushy tree is a much better example. What I've done is I've taken the artwork and I've added dark lines to come to the center, up to the top, this is the present now. We turned it upside down compared to the other ones. But if you look, let's say we dug down in the earth and we dug to this level of time and we looked across here and we found some fossils. What is the likelihood that the fossils we found are related to a living animal? Really, really low. Let's say this is humans and we follow that back down here and it goes here and then attaches to that. Well, we're looking here. What are we going to find? We would be extraordinarily lucky to find this. Look at all the other lines that are crossing that red line. If we found something over here, it's not related to our actual lineage directly, but it's a cousin. And at this point, if it's close, if we got one here or one here, it's very close in development to the human lineage. So that when you hear about Lucy or the something boy or the Flores people, what you're looking at there, if it's far up back in time, are things that might have some relationship to us, but it's not direct, and that's really important for you to really wrap your head around. And I wanted to be sure that you understood that the fossil record is better than it was, and people often simplify it in diagrams to make a point, but in reality, it's a big, messy bush, which looks like that. Will you stay?
That's okay, we can stay. We can stay.
Oh, Lord. Okay, last time, I want to review quickly again for you. Last time we talked about DNA, that it is in a double helix, that the importance of that is that one side reflects the other, is complementary to the other, that there are proteins that will unzip the DNA so that it can be read, and other proteins which do the actual reading and attaching new complementary nucleotides to the exposed DNA, thus causing a copy to be made on both sides. This is an enlarged diagram of DNA. This is the backbone with phosphates and the deoxyribose. And these green and purple, or mauve, purple and red, green and purple and so on, those are the nucleotides, and you can see the dotted lines between the oxygen and the nitrogen, just as I showed over here in one of my diagrams. So there's a hydrogen bond here and here, 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 and here. So this is a strong connection. And again, there are only 4 nucleotide bases, A, T, G, and C, or A, U, G, and C, and they're paired A with T or U, and G with C. And the opposite is true, C with G, T with A, U with A.